Welcome back, everyone, to our final session, which will be the lightning talks. So each speaker will have 3 minutes to talk, and note that there will be no individual question time for each speaker. So please submit your questions through the Q&A function throughout the talks, and those questions will all be asked together at the end of the session. Just remember to include the name of the presenter that you are asking the question for so that the chair can direct it to the correct person. And this session will be chaired by Dr. Jackie Chong and Dr. Huday Khalil. Chairs, over to you.
Thank you very much, Kalani. Welcome, everyone. And Kalani has so eloquently said what I was rehearsing to say, so I don't need to say it anymore. So these talks are for 3 minutes, and I guess without further ado, I will introduce our first speaker, who is Andy Nguyen. And Andy is a PhD student from Australia. And today he is going to talk about the prediction of antimicrobial resistance in Pseudomonas aeruginosa using integration of multiple genomic representations and graph neural networks. Thank you, Andy.
Thank you for introducing me. Hi, everybody. I'm Andy. I'm a first-year PhD student working under the library supervisions of Dr. Nenad Makesik. Today, I will be presenting our work on using machine learning with whole genome sequencing data to predict AMR in Pseudomonas aeruginosa. Pseudomonas aeruginosa is a particularly challenging pathogen because its AMR mechanisms are complex and often involve multiple interacting factors. Traditional rule-based approaches, which may look for the presence of sense of non-resistant genes, often fail short when dealing with Pseudomonas. And they can account for novel resistant mechanism or the complex interactions between multiple factors. This is where machine learning comes in. In recent years, it's emerged as a promising approach, successfully predicting AMR in various bacterial pathogens. Our goal was to develop a machine learning approach that could predict AMR to a wide range of anti-Pseudomonas strains. One of the key challenges we faced was how to represent genomic data. When working with the whole genome sequencing data, there are several ways to represent bacterial genomes and features for machine learning models. Certainly, there's no consensus on which representation works best. And we believe that by combining different representations might give us a more complete picture. And though this hasn't been widely explored in various research, and therefore we developed a novel approach using a graph neural network, which we call AMR-GNN. And the figures on the slide illustrates the high-level workflow of the studies. So first, we extracted 3 common genomic representations, the unit, the single nucleotide polymorphisms, and the frequency scatter games representations. After evaluating the predictive performance of each representation individually, we found Unitix perform best. And instead of simply combining all features into one vector, which could lead to overfitting, we built a graph neural network where Unitix served as the main node features, and with the other representations having to establish connections between isolates. And then with this AMR-GNN, we achieved significantly higher AUC compared to models using single genomic representations, particularly for fluoroquinolone and aminoglycosides, where models consistently perform well for all tested antimalarials. And then to address the black box issues of deep learning models, we also conducted model interpretations to identify which features contributed most to the predictions. And then this led us to identify several canonical AMR genes for levofloxacin and also tapromacin. When we compare wild type and mutants isolates of these genes, we observe significantly increase in AMT values in the mutants isolate, and therefore strengthening our findings. And so thank you for your listening.
All right, thanks, Andy. And next we have Muhammad Ridwan, who is a PhD student from Indonesia. Muhammad is going to talk to us about isolation of anti-MRSA metabolite producing bacteria from aquacultured and captive corals in Indonesia. Over to you, Muhammad.
Okay, thank you very much for the introduction. Okay, and so in this research that was led by my supervisor, who's a PhD in veterinary medicine faculty of OKM, and in his research, the aim of the study was to know whether or not we could actually isolate bacteria that produce secondary metabolites with anti-MRSA activity from captive corals. So previous studies have done with wild corals, and they could retrieve up to 41% of the bacteria from microbiome itself, possessing some degree of antibacterial activity, one of them being anti-MRSA. But actually, up to 99% of coral microbiome bacteria is uncultivable, meaning that if we were to isolate from white corals, only about 41% of those 1% actually possess some antimicrobial activities. So we figured that whether or not we could actually keep captive corals as kind of like genomic resources in our laboratory instead of actively isolating from wild corals, which require more resources. In these methods, we steril wash the corals with artificial salt water. We then isolated the bacteria from microbiome using T-Strict method with marine 2216 agar at 28 degree of Celsius for 2 weeks, and then we pre-screen the antibacterial activity against MRSA with this diffusion, and then when we got the 4 strains of with anti-MRSA activity, we then split one isolate into 2 culture, one of it with which containing the collagen scaffold as a 3D structure, and the other one in just liquid culture, and then we again I incubated for 1 week, and then we after that do another distribution and also MIC with serial dilution. And from the result, you can see from the first image that the our 4 isolates actually possess a significant antibacterial. And in this research, it's important to note that the collagen scaffold itself actually alter the growth of the bacteria strain, and they reach the stationary phase way faster. And it could be found out that up to 38.9% of those bacteria that could be cultivated with the addition of coral juice could not actually be further cultivated, and the coral juice itself actually directly contributed to the survivability of the coral microbiome bacteria. And the conclusion is that first, the captive corals contain even though lower than wild corals, a lot of anti-MRSA secondary metabolite producing bacteria. Thank you, and hope we could have further discussion at the Q&A. Thank you.
Thank you very much, Mohammed. Our next speaker is Arijit Nandi, who is a PhD student from Australia. And Arijit is going to be talking about seeing the unseen, imaging bacterial infections. Thank you very much, Arijit.
Good afternoon, everyone. Today I'm presenting my PhD topic, which is entitled Seeing the Unseen, Imaging Bacterial Infection. As you all know, bacterial infection, especially those caused by antibiotic-resistant organisms, pose a significant global threat to human health. So there are traditional diagnostics which are often used, but it is unable to detect deep infection, such as prosthetic joint infection. And there is an urgent need to enhance diagnostic methods to avoid unnecessary surgery and improve patient outcomes. So basically, I am developing some new diagnostic probes rather than the traditional inhibitors. So previously, there are traditional approaches in infection diagnosis like microbiology, surgery, sequencing, but this all requires a different type of blood or urine samples and sometimes the invasion. So it has a lot of disadvantages. So recent techniques like imaging based diagnosis, which is quite popular in cancer diagnosis and anti-cancer drug development. So it has a lot of advantages, like it is non-invasive, faster than culturing, gives spatial information, temporal monitoring of infection progress and treatment. But it also had a lot of challenges nowadays because it can't specify the host cell and the targeted cell. It can specify the inflamed tissue with the infection site, and obviously the circulation time, because most of the imaging based diagnosis are based on the radioisotopes. So it should be eliminated from your body within a certain period, and it should give the proper activity also. So based on these advantages and disadvantages, we designed our new diagnostic tracers, which will bind on the bacterial surface. And for the surface binding, we took different type of antibiotics for gram-positive as well as gram-negative. And we are adding different type of linkers. It will be flexible or rigid linker for the better solubility and the permeability. And we are adding one chelator. So we will add different type of chelator for the optimization, which is the best one to hold the radioisotope. And the last one of our design molecule is the radioisotope, obviously, because we are going to check the PET imaging of our molecules. We will do the in vitro and in vivo also. Proposed outcomes and significance. So this kind of diagnostic tracers can lead to precision medicine development, and we can understand the pathogenesis and diagnosis rather than treating the AMR blindly. And we can diagnose the gram-positive as well as gram-negative. Thank you. Thank you for giving me the opportunity. Yeah.
Thanks, Arijit, for your talk. Next, we have Shalini Roy, who is a master student from India. Shalini is going to talk to us about using CRISPR-Cas9 to study correlation of quorum sensing and virulence in Pseudomonas aeruginosa. Over to you, Shalini.
Hello, everyone. Am I audible? Thanks for the introduction. I'm Shalini. I'm here presenting my small section of my dissertation work. Starting with introduction. As we know, Pseudomonas aeruginosa is gram-negative ubiquitous human opportunistic pathogen, which is capable of causing wide array of chronic infection. There are 4 major pathways present in Pseudomonas, where LASA reside at the top of the cascade, which is also responsible for the production of various virulence factor. Here we are mainly targeting LASA gene using CRISPR, which allow efficient and scarless genetic manipulation in Pseudomonas. And this is the well-known mechanism of cytosine-based editing, which converts cytidine to thymidine using cytidine deaminase, and also capable of inactivating targeted gene by generating premature stop codon. Coming to the methodology part, plasmid extraction and confirmation was done initially, followed by spacer selection and phosphorylation. Here we have selected our targeted spacer using CRISPR CBI, which is one of the finest CRISPR tool available in the market. The ligation was done using Golden Gate cloning, and followed by transformation using electroporation, and then subculturing virulence factor assay, lastly plasmid curing. Let's talk about the result. The first gel image is showing the extracted plasmid PN-Caspar-Bake, and the second gel image is showing the linearized plasmid after digestion with EcoR1. After transformation, we got the significant number of resistant colony on xanthomycin plate. But once the confirmation was done, we have performed virulence factor assay for protease as well as exotoxin A. And we found that in case of P1.14 variant, there is a reduction in significant amount of secretion in comparison to wild type P01 in both the cases, in protease as well as exotoxin A. We also have done Sanger sequencing and PCR to confirm our base editing. But unfortunately, in that case, we didn't get the result we expected. The base editing was taking place in that region only, this yellow read part. But somehow it, due to off-target mutation, the base editing took place from 160th position away from the targeted area. Also have done plasmid curing to confirm the plasmid deletion. Here we grew our variant in sucrose medium in presence and absence of antibiotic containing media, where we can see that there is proper growth without antibiotic media, and there is no growth the media containing xanthomycin, which is indicating the completely curing the plasmid. So overall, we can conclude that the inactivation of the LASA significantly impairs the production of virulence factor in Pseudomonas aeruginosa, such as protease and exotoxin A. I would like to thank the DBT funding program and Maharaja Sayajirao University of Baroda, and last but not the least, Monash University for giving us such amazing opportunity and for the lovely talk. Thank you.
Thank you very much, Shalini. Our next speaker is Afsaneh Ashadi Edlo, and today Afsaneh is talking to us about harnessing the body's natural defenses, dual function antibiotics, and synthetic biology to combat antimicrobial resistance. And I should also add that Afsaneh is a master's student from Iran. Thank you.
Thank you very much. Today I want to talk about two innovative approaches. By inspiring the way human body naturally fights infection using molecules, cortisol, serotonin, and antibodies, I asked, can we develop a smarter antibiotic that works in both therapy and diagnosis while reduce resistance? And this led me to explore two cutting-edge solutions: dual-function antibiotic and synthetic biology. Dual-function antibiotic have diagnosis and therapeutic properties at the same time. Nowadays, we use fluoro antibiotic, but this kind of material have some disadvantages such as low penetration and off-target effect and short fluorescence lifespan. So I suggest new materials, multifunction materials. Metal organic framework quantum dots composite have some advantages such as controlled drug release and target detection, in real-time imaging, and also they can have lower resistance development. And with this method, we can study resistance mechanisms and also treat infection at the same time. And this can be an amazing achievement in antibiotic research. But this is not enough, and we need new antibiotics. Synthetic biology can change the game in this way. Throughout the synthetic biology, we can train bacteria and microorganisms to produce polyketids and non-ribosomal peptides. But we have a problem. Many microbes have silent genes, and we need to active this kind of active silent antibiotic gene. For this purpose, I suggest to use a stress condition activation throughout nutrition limitation, metal exposure, and also pH shift. Also, we can directly using CRISPR-Cas9 activation method for active this silent genes and produce new antibacterial and new antibiotics for our purpose. And we please consider that with this method, we can create new antibiotic, and we don't need to discover a new antibiotic. And I think that it's really important to do more research like this, and our future start with investing in new ideas today. Thank you very much.
Thank you, Afsaneh, for your talk. Our next speaker is Ormi, who is a PhD student from Australia. She's going to talk to us about construction of antiviral surfaces using peptidomimetics. Over to you, Ormi.
Thanks for the introduction. Good afternoon, everyone. I'm delighted to present my work on the construction of antiviral surfaces. This work is conducted at School of Optometry and Vision Science at UNSW. My PhD research is focused on antiviral compound, but I believe today's topic, constructing antiviral surfaces, is highly relevant to this symposium. Just as virus can persist on surfaces and act as a reservoir for infection, other antimicrobial resistant microbes, mainly bacteria, can also spread easily and leading to nosocomial infections. While traditional antimicrobial disinfectant are effective, but they require continuous reapplication. To address this limitation, we aim to develop surfaces with inherent antimicrobial properties. For this, we used peptidomimetic 758. One of my colleagues, Sham, already talked about this peptidomimetic against Candida aureus, and I am presenting its effect against viruses, a compound which is developed by our group and tests antiviral efficacy against coronavirus and influenza virus. For surface coating, we employed a simple one-part method using dopamine as an adhesive element. The first step was to confirm the successful attachment of this peptidomimetic to the surface, which we confirmed using XPS and TOF-SIMS spectrometry, detecting elemental and ionic compositions. We also conducted water contact angle measurement and observed that 758 coated surfaces became more hydrophobic than their control. For antiviral assessment, we followed ISO standard and found that 758 coated surfaces significantly reduced both virus. In parallel, for any antiviral test, it's important to test the cytotoxicity as well. So we confirmed the cytotoxicity profiling of this coated surface and against 4 different cell lines. And we found that coated surface are non-toxic. In conclusion, 758 coated surface demonstrated strong antiviral activity while maintaining biocompatibility. So we believe that peptide and peptide mimics can open a new avenue and for their further application to fight against AMR and other antimicrobial options. However, before real-world application, further studies are needed to expand microbial testing beyond viruses, like including other microbes, and also we need to expand the range of viruses before declaring it as a broad spectrum compound. And also we need to expand this in vitro assays to in vivo and ex vivo evaluation for both antimicrobial perspective and cytotoxicity profiling. Thank you for your attention. I'm happy to take or discuss anything after our talk.
Thank you very much, Urmi. Our next speaker is Rahul Maitra, who is a PhD student from India. And today, Rahul is going to talk about the synthesis of tryptanthan appended dispiropyrrolidine oxindoles and their antibacterial evaluation. Thank you very much, Rahul. Sorry, we can't hear anything. I think your microphone is on mute.
Hello, am I audible?
Yes, we can hear you now.
Okay, thank you, ma'am. As we all aware about antimicrobial resistance, which is also well known as a silent pandemic. Today, I am focusing on Staphylococcus aureus, which can cause millions of deaths worldwide. WHO declared methicillin-resistant Staphylococcus aureus as a high priority pathogen due to its widespread multi-drug resistant property. So the problem is critical due to the unavailability of treatment options. So to fulfill the unmet need, we mainly design the tryptophan pyropyrrolidine oxyindole hybrid molecules and evaluate their antimicrobial properties against the Staphylococcus aureus. As we've seen that from a series of compounds, mainly 3 compounds have been shown, MIC less than 0.25, that is 0.125, and after that we have evaluated the cytotoxicity of those compounds in the Vero cell line. This is the African green monkey kidney cell line, also is a mammalian cell line, and we have found that the selectivity index of those compounds of 2 compounds is greater than 80 and the 1 compound is 40. We further proceed with those 3 compounds and check the MIC of those 3 compounds against the multi-drug resistant Staphylococcus aureus clinical strains, and they are also multi-drug resistant strains, and we have found that the 2 compounds 5B and 5C has shown equipotent activity against the multi-drug resistant Staphylococcus aureus strains. And after that, we have chosen the 5B compounds and check the combination of 5B with the FDA approved drugs against Staphylococcus aureus, and we have shown that our compound 5D has not shown any interaction with the FDA approved drugs. So maybe our compound 5B has a unique mode of action. And thank you, ma'am.
Okay, thank you, Raul, for your talk. Our next speaker is Verika Bhatt. She's a PhD student from Malaysia. Verika is going to talk to us about antibiotic resistance knowledge in Malaysia. Over to you, Verika.
From University of Malaya, Malaysia. Now, as we all know, AMR is a global health crisis, and if the current trends continue to rise, the Western Pacific region alone is projected to see 5.2 million deaths by 2030, and that's merely 5 years away. A major driver of AMR is the misuse of antibiotics, often caused by public misconceptions about their use. To address this, our study aimed to assess the level of knowledge about antibiotics and antibiotic resistance among Malaysians. We conducted a nationwide survey from April 2022 to March 2023 using a WHO-approved questionnaire via Google Forms and in-person interview. A total of 517 participants were recruited. The majority of participants were females, aged 30 to 49 years old, and from central Malaysia. Most had tertiary education and lived in urban areas. As for antibiotic use, a majority, 98.5% of participants had used antibiotics, and many had misconceptions about their use. A significant number believed that antibiotics could treat sore throat, fever, colds, flu, which are all very viral infections. As a matter of fact, only 41.2% had low knowledge on antibiotic usage, and 36% had low knowledge on AMR. Regression analysis showed that age, education, and income were positively associated with better knowledge of antibiotics and resistance. Now, I would just take a moment to note that the age group of 18 to 29 years old, youths just like us, were found to have the least knowledge among the rest of all, which comes as a shock to me because I feel like we have information at the tip of our fingers at all times. So we did do further analysis, a correlation analysis that found that those that had better knowledge of antibiotics had better knowledge of antibiotic resistance as well. Nonetheless, the correlation was still little, which does tell us something that we need an intervention to improve this. In conclusion, while many Malaysians have used antibiotics, have a knowledge and understanding on how to use it, there's a lot of knowledge gaps that remain and misconceptions that persist. So addressing these through education is very crucial, and we definitely need awareness programs to engage younger age groups from 18 to 29 and lower socioeconomic groups. We also require more qualitative research to identify what the underlying causes of inadequate knowledge are. I'll just like to end this by saying that I have a QR code at the bottom. I'm doing a podcast on AMR awareness, a multidisciplinary approach. So if you're interested, you can take a look. I would like to also acknowledge the University Malaya Scholarship Scheme for supporting this study, and thank you for your time.
Thank you so much for that, Vedika. Our next speaker is Dipanshi Saxena, who is a PhD student from India. And today we are going to hear about, Pivinium pamoate, how it potentiates levofloxacin against levofloxacin-resistant Staph aureus. Thank you very much.
First of all, am I audible? Okay. Thank you for the introduction. I'm Dipanshi Saxena from CSIR CTRI, and here I will be presenting my work on repurposing an anti-helminthic drug as antibacterial. We have screened the Pivinium palmoate against a panel of gram-positive and gram-negative bacteria, where we found that Pivinium palmoate was selectively targeting gram-positive Staphylococcus aureus with MIC as low as 0.5 microgram per ml. Additionally, it was not found cytotoxic against Vero cell lines. Next, we were curious to know whether it has the activity against multi-drug resistant isolates also. So we screened it against a panel of MRSA and VRSA strains and found that it was equivalently inhibiting the growth of multi-drug resistant clinical isolates of Staphylococcus aureus. Next, we were curious to know whether it is active, the Pivinium palmoate is active only against the planktonic bacteria or also against the biofilm forming the cells in biofilms and the bacterial cells which are intracellular. As seen from the first two graphs, we can see that it was potentially inhibiting the growth of bacteria in biofilm also and also in intracellular bacteria. Next, we were curious to know the mode of action of Pivinium palmoate, whether it is acting as bactericidal or bacteriostatic. From the first graph, we can see that the drug is bactericidal with a reduction of around 6 logs within the first hour of compound treatment, as seen from the first graph, and it was more effective than the standard drug vancomycin. Secondly, it was found potentiating levofloxacin against the Staphylococcus aureus at 1x MIC, when, as we can see from the graph, that both the combination of the two drugs are having the potency of reducing the bacterial load after 24 hours of drug treatment. Next, we question whether this in vitro activity of the Pavinium palmoid can be translated in vivo. So we checked it in the murine model, thigh infection model of infection, and from the first graph we can see that the Pavinium palmoid showed a reduction of around 1.5 log in the bacterial load and also potentiated levofloxacin against levofloxacin resistant Staphylococcus aureus strain. And from the second graph, we can see that it is showing 0.5 log reduction in the bacterial growth. Thus, we can say that repurposing is a quicker and effective approach of drug discovery. And secondly, Pavinium palmoid has the potential of studying further as an antibacterial agent against Staphylococcus aureus. I acknowledge my institute, CSIR CTRI, for giving me the platform of performing the work, my supervisor, Dr. Siddharth Chopra, for aiding and guiding me through the work. And thank you all for your patience.
All right, thank you, Dipanji, for your talk. Our next speaker is Sulaiman Arfan. He is a PhD student from Australia. Sulaiman is going to talk to us about susceptibilities of Pseudomonas aeruginosa to antibiotics and bacteriophages. Over to you, Sulaiman.
Hi, everybody. Thank you very much for providing us opportunity to present our research. So my topic is comparison of susceptibilities of Pseudomonas aeruginosa to antibiotics and bacteriophages. First of all, Pseudomonas causes a microbial keratitis. This is a disease of the eye, basically inflammation of the cornea, which is caused by bacteria, fungus, and viruses. Among all these pathogens, Staph aureus and Pseudomonas aeruginosa are two most common pathogens causing microbial keratitis worldwide. It is an inflammation of the cornea, which can lead to a loss of the eye if left untreated. There were so many cases in Australia, and 2 years ago, 4 people leads to loss of death, 4 people results in their loss of eye, and 14 other patients results in the loss of vision. So there are so many antibiotics which are used to treat the microbial keratitis, but due to acquired resistance and some horizontal gene transfer mechanisms in Pseudomonas aeruginosa, these antibiotics fail to treat microbial keratitis cases or patients. So bacteriophages offer a promising alternative solution to treat microbial keratitis. So the objective of this study is to evaluate the susceptibilities of this bacterium to different antibiotics and bacteriophages, and to determine the association between bacteriophages and antibiotics. So these are my results and discussion. These are ciprofloxacin and levofloxacin, which are fluoroquinolones and other aminoglycosides group, gentamicin and tobramycin, and their cutoff values by CLSI, their standard intermediate resistance. And another one, if we go to antibiotic susceptibility rates and fast sensitivities, out of 62 isolates, ciprofloxacin showed 63%, levo 80.6%, gentamicin in 95, followed by tobramycin, which is 76%. And there are 6 phases which are used, 56.4. They all showed their percentages. So table number 3 shows the association between susceptibility to antibiotics and phages. So this all research and study demonstrated that all phages were active against Pseudomonas aeruginosa, with DSU4 being the most effective one among, shows the 74% among the other phages. And the fluoroquinolone antibiotics, gentamicin and levofloxacin remain effective antibiotics to treat Pseudomonas aeruginosa keratitis in Australia and India as compared to aminoglycoside antibiotics. So, a significant positive correlation was found between by using the Pearson correlation test to determine their correlation values between the relationship of all phages to different types of antibiotics. So as we found that a positive correlation was found between only 2 phages, DSU2 and DSU6, found between the fluoroquinolone antibiotics, which shows that there is a positive correlation. If sensitive shows in antibiotics, then these isolates were also sensitive to phages. So DSU2 and DSU6 only showed the positive correlation. If they show sensitive to these 2 phages, then these isolates show sensitivity to antibiotics as well. So this is all from my side. And again, thank you so much for the organizing committee to arrange such a Indo-Pacific AMR symposium for us to show our research. Thank you so much.
Thank you very much, Suleiman. Our next speaker is Shakil Shahzad from Australia, and he is a PhD student. And today he is going to tell us about the patterns of susceptibility of clinical isolates of Acinetobacter towards polymyxins and various cationic antimicrobials. Thank you.
Hello, everyone. Thank you so much for providing me the opportunity. So Acinetobacter is gram-negative Coccobacillus, basically associated with nosocomial infections. And Acinetobacter baumannii is one of the most important pathogen that is reported by WHO as a priority pathogen that is resistant to all known antibiotics as a pandemic, drug resistant microorganism. So here the aim of study was to evaluate the pattern of susceptibility to see if the polymyxin resistant strains are also resistant to other cationic antimicrobials. So we use Malfor antimicrobial peptide and melamine and LL37, which is the natural antimicrobial peptide, and lactoferricin. So here we did the susceptibility testing. In first group, you can see the strain of Acinetobacter very sensitive to polymyxin and cholesterol, as well as they show cross sensitivity to the cationic antimicrobial peptides. While strain Acino10 show resistant to polymyxin and also cross resistant to other cationic antimicrobial peptides. Strain A2 follows the same pattern, same A9 in the same pattern, but interestingly, the strain that is used in the last, that is the ATCC strain, mutant strain, highly resistant to polymyxin, both of polymyxin B and cholesterol. That strain show sensitivity to Malfor, and comparatively to melamine is show the minimum inhibitory concentration lower than the melamine, that is the main peptide, while the Malfor is the synthetic peptide, which is the smaller in sequence of amino acid, as well as it also lack specific amino acid that are associated with membrane binding. But in case of mutant strain, this peptide Malfor is very active against polymyxin resistant strain. We show that the Malfor may have the potential to bind the target other than the membrane of the bacterial cells. And interestingly, in our result so far, we found that the strain that we are searching for cross-resistant is, in case of Acinetobacter, strain are not showing the cross-resistance in a regular pattern. There is a reverse pattern of susceptibility, and this is the conclusion which suggests that there is a distinct mechanism of action may be followed by the peptide like Malfor against Acinetobacter, and moreover, the mechanism of action is independent of LPS binding, so that the MIC is quite lower than the other compound in case of, particularly in case of Malfor and LL37. So in conclusion, there is a diverse pattern of susceptibility and distinct mechanism of susceptibility followed by the different organism of the isolate of Acinetobacter baumannii and Acinetobacter species isolated from clinical samples. This is all from my results. Thank you so much for providing me the opportunity to talk in this platform. Thank you.
Thank you, Shakil. Our next speaker is Jawei Shin. She's a PhD student from Australia. She's going to talk to us about whole genome sequencing-based prediction of antibiotic resistance in ocular S. aureus across 6 continents. Over to you, Joey.
Thanks for the introduction. So Staph aureus is a leading cause of ocular infection, and which can result in the vision loss and blindness in the several cases. And the treatment for the ocular infection is typically relying on the antibiotics. However, as we know, the spread of the AMR has become a growing concern in the management of the ocular infection. So this study aims to review the antibiotic resistant profiles of ocular resistance profiles of the Staph aureus. So what did we do? We have downloaded all the available and assembling whole genome sequence data from the NCBI regarding the Staph aureus, which are isolated from the patients with the ocular infection. So there are around 195 biosamples have been included, and then the card and the responder have been used for further analysis for the resistant genes and prediction of the resistant phenotypes, as well as a comparison of the resistant prevalence. So we can see the results here. There are a total of 40 resistant genes have been identified, and we can see the prevalence of African palm genes is quite high. Besides, the beta-lactamase gene showed a high prevalence around 800%. Sorry, 8%. And also the resistance to 45 antibiotics have been predicted. So from the heat map, it's easy to say that the isolates from the North America and Asia showed the most extensive resistant profiles, while the isolates from the Oceania showed the lowest resistant spectral. So, and by comparison, we found that the resistant prevalence between the continents vary a lot. So in this table, I just put the p-value here. The yellow one means the value higher than the global level, and the blue one means lower than the global level. So you can see the North American isolates are more resistant to the MLSB antibiotics, whereas the isolates from the Europe and Oceania showed the opposite trend. Besides, both North American and Asian isolates showed more resistant to the aminoglycoside antibiotics. On the other hand, the Oceania isolates are less resistant to the beta-lactam antibiotics. So in conclusion, this study just gives us some critical information that could help us to customize the treatment of the ocular infection. Yeah, that's it. Thanks for the listening and passion.
Thank you very much, Jiawei. Our next speaker is Lakshmi, who is a PhD student from India. And today, Lakshmi is going to talk to us about phage-based solutions for UPEC infections in terms of investigating resistance mechanisms and therapeutic potential. Over to you, Lakshmi.
Thank you for the introduction, and good afternoon, everyone. I'm Kavya, and I'll be sharing a brief overview of my research project focusing on combating antibiotic-resistant UPEC using phage therapy. So imagine suffering from a urinary tract infection that keeps coming back, each time harder to treat. And UTIs affect the daily lives of 150 million individuals every year. Uropathogenic E. coli, or UPEC, is a primary causative agent responsible for over 90% of the community-acquired cases of UTI. UPEC is a pathogen notorious for its ability to form biofilms and evade antibiotics, leading to persistent infections. With antibiotic resistance on the rise and new drugs scarce, treating UTIs is becoming increasingly difficult. This underscores the need for alternative solutions. My research focuses on bacteriophages, viruses that precisely target and eliminate bacteria. Unlike traditional antibiotics, phages are highly targeted and don't harm or induce resistance development in off-target bacteria. Moreover, the specificity of phages allow them to target key virulence pathways in UPEC. While phage therapy offers great potential, bacteria, including UPEC, can evolve resistance to phages, limiting their long-term effectiveness.
To develop a sustainable phage-based treatment, we need to understand how UPEC interacts with phages and how we can potentially overcome resistance. And the first step to do so is to isolate phages that can infect UPEC. We have collected environmental water samples and screened them for naturally occurring phages. The isolation part is now complete, and we have a collection of phages that can target UPEC strains and are currently working on characterizing them. But even when phages work, UPEC can develop resistance to them. This brings us to the second question. What genetic changes allow UPEC to resist phages? I plan to use mutant libraries and whole genome sequencing to pinpoint the genes responsible for phage resistance and understand these resistance mechanism. Finally, we want to find out if we can enhance UPEC's susceptibility to phages. If we can find ways to reverse or suppress phage resistance, we can make phage therapy more effective. One, via genetic modulation, I plan to knock out resistance genes to confirm that the mutations present are to see if the UPEC strains become more vulnerable again. Second, via chemical modulation, I plan to screen compounds via in silico and in vitro tests to find ones that increase vulnerability of UPEC to phage attacks. Ultimately, phage therapy could be a game changer in treating antibiotic-resistant UTIs. By identifying potent phages, uncovering resistance mechanisms, and exploring ways to enhance susceptibility, we hope our research could contribute to the development of sustainable phage-based treatment for recurrent UTIs, reducing our reliance on antibiotics. Thank you.
Thank you, Kavya, for that. Our next speaker is Bhavya Mishra. She is a master student from India. The talk is going to be about the impact of BRLR activation by SAG-S on antibiotic resistance profiles in biofilm-forming strains of Pseudomonas aeruginosa. Over to you, Bhavya.
Thank you for the introduction. So I'll straight jump up to the introduction. So basically SAG-S is a transmembrane protein that is present in Pseudomonas, and it's get activated by some environmental cues such as bacterial attachment. And upon activation, SAG-S causes the regulation of secondary messenger, which is cyclic di-GMP. And as the level of cyclic di-GMP increases, BRLR, which is biofilm regulator locus, it gets activated. And upon activation, it causes the biofilm formation, enhanced biofilm tolerance, and multi-drug resistant in Pseudomonas. So our hypothesis is to study biofilm-mediated antibiotic resistance through the activation of BRLR. So the methodology that we followed was whole genome sequence data analysis, MIC, then biofilm quantification, AST, and piocynin quantification, followed by gene expression studies. So we selected two strains, which is TP10 and TP11, because they were biofilm forming. And we did their whole genome sequence data analysis, and we found that there were few silent and synonymous mutation in efflux pump and regulator genes, while in the TP10 there was truncated SAG-S, and in TP11 it was there was a deletion mutation in the NALD regulator. So the results were as followed. The ongoing MIC, we found that TP10 was susceptible to both the antibiotic, TP11 was resistant. And biofilm quantification showed that TP11 was a very high biofilm producer and TP10 was a very weak biofilm producer. The biofilm AST showed no significant results among three, like PA01 was a reference strain, but there was no significant difference in TP10 and TP11. Piocynin quantification showed that there was the TP11 strain was very high piocynin producer, while TP10 was a weak piocynin producer. And upon doing the gene expression studies, we found that the SAG-S, BRLR, MEX-A efflux pump, and MEX-F efflux pump were highly upregulated in TP11, whereas the NALD and NALC was downregulated in TP11 as compared to TP10. So we concluded that that as TP10 was a susceptible strain, the mutated SAG-S caused less expression of SAG-S and BRLR, and hence less piocynin production. Whereas in TP11, as it had mutated NALD, which is negative regulator of efflux pump, there was a highly overexpression of SAG-S, BRLR, and efflux pump, and hence high piocynin production. So that was all it. I acknowledge my teaching staff at MSU and DBT for funding. Thank you so much.
Thank you very much, Bhavya. And our final speaker, but certainly not least, is Mahi Raj. And Mahi is a master's student from Australia. And today, Mahi will tell us about megaplasmids and prophages, a deep dive into carbapenem-resistant Klebsiella pneumoniae genomics. Take it away.
Thank you for the introduction. Starting with the introduction, like the Klebsiella pneumoniae is known as a major escape pathogen known for causing most of the nosocomial infection, and it was earlier reported that Klebsiella is harboring most of the megaplasmid and prophase sequences in it, which help in disseminating the AMR gene. So our main objective was to perform the whole genome characterization of the Klebsiella genome. Starting with the methods, we did whole genome sequencing data analysis of the 16 isolate of the Klebsiella pneumoniae, followed by the megaplasmid isolation and then the induction of the profile. So our key finding was that most of the isolate of this Klebsiella belong to ST147, which was predominantly global clone of the Klebsiella pneumoniae. Then we found that the most prominent carbapenem resistant gene was the BL-NDM-fine OXA48-like, which is mostly located on the plasmid, whereas very less was present on the chromosome. Then we look for the replicon type. INC-F1 and INC-F2 was more predominant in all the clones of the isolate of the Klebsiella. Then we look for the other insertion element like SKP23, which is more common. Then this is the graphical representation. We saw the plasmid and AMR gene association, which represent that the most of the AMR gene is associated with the conjugated plasmid. Next, we isolated the megaplasmid, and we found that most of the isolate is showing the band more than 20 kV, which suggests that all the isolate is having the megaplasmid in it. Next, we are interested in the DJ strain, so we performed the prophase sequence analysis, and we found that DJ is having the 5 prophase sequence in it, out of which 4 were present on the chromosome and only 1 is present on the plasmid. Next, we induced the prophase with the mitomycin different concentration, and we found that as increase in the mitomycin concentration, the growth rate of the DJ is decreasing, and this mitomycin is causing the bacteriostatic effect rather than bactericidal. Then we use the sub-MIC concentration to induce the prophase, and we found that when we took the sub-MIC concentration, the prophase sequence which is present in the DJ strand, it is inducing. So to conclude overall this story, what we found that our isolate is having multiple megaplasmid and prophase sequences in it. And as to acknowledge this, I would like to thank the Gujarat State Biotechnology Mission and the DBT supporting teaching program for funding support, and Monash University for giving me this opportunity to present my piece of work. Thank you.
Thanks, Mahi. And this brings me to the end of this session. I would like to thank all the speakers for their fantastic talks and the audience for listening to us today. And we've got time for 2 to 3 questions. If you would like to ask any question, could you please let us know in the chat box now? And also please specify the speaker whom you're asking as well. Thank you.
I guess while we're waiting, perhaps I can ask a question. Let me see.
Yeah.
Okay, so I've got a question for Shakil. So you were analysing AB 19606, oh sorry, ATCC 19606. That's a fairly old clinical isolate. Have you looked to see whether your cationic peptides are active, or what kind of effect it would have on something more recent, like for example, AB 307 0294?
Currently we have this strain, but we are looking in future, we are looking for the further strains to evaluate. So thank you so much for the suggestion.
Thank you very much.
Yeah.
Anybody else got any questions? Okay, if not, I've got another one. Oh, unless you have one, Uday?
You go for it.
Okay. Let me see. Oh, yes. So Saxena, your presentation was really great. I was just wondering, do you know what the mechanism of action against Staph aureus is? Like the chemical that you're looking at, pyrovinium pamoate, what the mechanism of action that it has? And also, has this been tested in an animal model?
Ma'am, actually, we are working towards finding the mechanism of action of the pyrovinium palmate, but as anti-helminthic, it is disrupting the energy metabolism of the organism, of the pinworms, basically. It
Okay.
is known.
Okay. Thank you very much for that.
Okay.
Any other questions? No? Okay, I've got to do a deep dive into my questions then. Let me see. Oh, Lakshmi, great talk. I was just wondering, oh, sorry, Kavya, sorry, I'm very sorry.
Okay, anything works.
Sorry. So with some bacteria, when they develop phage resistance, they gain antibiotic sensitivity. So have you looked at that with your EPEC strains?
So I am actually in the very preliminary stages of my study. So when we do get our phage-resistant mutants, we will definitely look into it.
Thank you. We
You're
have
welcome.
a question.
Okay, we've got a question from Dipanshi to Shakil. Is the cytotoxicity checked for peptides against mammalian cells?
No, currently we haven't tested the cytotoxicity against mammalian cell. Particularly Malfor and melamine, I think they are already tested for cytotoxicity if you see the literature review. And LL37 is already human antimicrobial peptide, so they are not cytotoxic to the cell. Malfor already, our peptide Malfor already passed phase 4 clinical trial. So it's not really cytotoxic. Thank you.
Okay, I think we have one more question.
So, yeah.
Oh, so Rain Hussein has a question for Ridwan. Rain is asking whether you can, how you extract the metabolite from the corals, and do you compare it with known antibiotics against MRSA?
Okay, thank you. I'm sorry, I was having a network problem. So the extraction method was we centrifugate first, and then we extracted the pellet and the supernatant each to their own. We used methanol with solvent to extract a ratio of 1:2, and then we repeated it 3 times, and then we dry it using magnesium sulfate and hydrous magnesium sulfate, and then we rotafab and then just extract it into without the solvent out, and then we actually compare the what is it called, the inhibition zone with known antibiotics, but I'm not sure about the type because it was done by other researchers. So this study focused on the isolation and potential of captive coral itself as source of genetic resources for bioprospecting. So it doesn't not focus on the capacity of inhibition of the isolates that were extracted. And yeah, that's it. Thank you.
Thank you very much for your response. And I think that will sort of draw this session to a close. Thank you very much for to all the speakers and also to all the participants. So thank you very much for a very wonderful afternoon.
All right. We have now reached one of the most anticipated moments of our symposium, recognizing the remarkable contributions of our participants. Before we announce the winners, we want to take a moment to appreciate each and every one of you who participated in this symposium. We have seen incredible research and innovative ideas, and we truly appreciate everyone's efforts. Now, it's time to recognize some of the outstanding contributions made in each session. Please feel free to share your congratulations in the chat or use the reaction buttons. Session 1. Innovative Technologies and Interventions. The winner is Sophie Low from Monash University, Malaysia. Congratulations. The runner-up is Jamaya Hemphill from Hudson Institute, Monash University. Congratulations to you too. Session 2. Environmental, Economic and Social Impacts of AMR. The winner is Jia Chibe from Doherty Institute, University of Melbourne. Fantastic, congratulations. Session 3. Regional AMR Challenges and Solutions. The winner is Tanzina Akhtar from University of New South Wales. Congratulations. Runner-up, Shyam Kumar Mishra from University of New South Wales. Good job. Session 4, Lightning Talks. The winner is Vedika Bhatt from University of Malaya. The runner-up goes to Bhavya Mishra from the Maharaja Sayajirao University of Baroda. Congratulations to you both. In addition to the session awards, we also want to recognize 2 participants who asked the most insightful questions during the symposium. The Best Question Awards go to Abhinaba Ray from Monash University and Rain Hussein from University Kebangsaan, Malaysia. Congratulations to you both. Thank you to everyone who actively participated in the discussions. Your questions made this symposium even more engaging and meaningful. A quick reminder to all our winners, please send your address and contact information to the organizing committee via email so that your prizes can be sent to your preferred location. The email address has been shared in the chat. A huge congratulations once again to all our winners and a heartfelt thank you to all participants for making this symposium such a success. We would also like to extend our sincere gratitude to the Center to Impact AMR and CSIRO for the incredible support in making this event possible. Our acknowledgements go to Professor Anton Peleg for opening the symposium for us. A heartfelt thank you to our keynote speakers, Professor Mark Blaskovich and Professor Trevor Lithgow, who has graciously given their time to share the cutting-edge research undertaken by their labs. We would also like to extend a massive thank you to Veronica Lay from the Center to Impact AMR, who worked tirelessly to ensure this event ran smoothly. To all our speakers and attendees, thank you for sharing your knowledge, engaging in thought-provoking discussions, and making this symposium a platform for meaningful collaboration across the Indo-Pacific region. The conference has brought together over 100 participants registered from 8 countries, 17 research institutes, and 34 centers for learning and discovery. We hope this symposium has not only enriched your academic journey, but also inspired you to continue working together to push the boundaries of innovation and research. Before we wrap up, please take a moment to fill our event feedback survey. You can scan the QR code here, and your input will help us to improve future events. And also, if you'd like to be involved in organizing the next Indo-Pacific Future Leaders Against AMR Symposium, email us at the email address in the Zoom chat. Also, I'd like to remind everyone about our networking session on GatherTown, which is a great opportunity to connect, discuss ideas, and continue the conversations from today. GatherTown link is also in the chat, or else you can scan the QR code here to join. Our committee will be here to assist, and if you need help, you can email us or rejoin this Zoom meeting for support. The networking session will remain open until 7 p.m. AEDT, so make the most of it. With that, we conclude the inaugural Indo-Pacific Future Leaders in AMR Student Symposium 2025. Thank you once again, and we hope you enjoy the networking session.